Here's the deal. We have a lot more information to go over today, and the information today is a little bit, it's complicated. And I was sitting there thinking about it, talking to my husband last night, and I said, you know, I think there's going to be no opportunity for more point value on this third exam. So you guys have to do very well on this exam. Does everyone understand? You have to do well. I've given you, well actually Michelle wrote up the review for the first part of the notes on this, but not for the remainder of them. So what I'm thinking is we'll finish up this information today, but I'm going to postpone the exam until after Thanksgiving. That will give you your Thanksgiving break to sort of sit down once in a while, look over your notes, and as long as that's okay with everyone. Does anyone have a problem with that? Anyone at all? And so then on Tuesday, then Michelle can do a review for that exam. I'm talking after this weekend, Tuesday, our next lecture, and that would be open for everyone. Everyone can come to that recitation and have a review for exam 3. Then, I know it's hard to see this, but your exam will be December 2nd, exam 3, and then Thursday of that week, Michelle's going to run a review for the final, and I will try and get the final done ahead of time so she has a copy of it so she knows kind of where to focus on the review. And then your final is Tuesday, December 9th from 8 until 10. Okay, everyone good with that? So have you all got those dates written down? And of course, all of you are welcome to come to both reviews. Yes?
Are we going to be in this class from now on?
We are going to be in this classroom from now on. All right? Now, yes.
It's final multiple choice.
Final is 101 multiple choice questions. Yes, it is. Now, the last thing that I wanted to say, oh yeah, I remember now what it is. I will be having a lecture on Tuesday. We should finish the information for the third exam today. So we will be covering just a few interesting topics for the remainder of the semester, and maybe get into a little bit of population genetics and cancer genes and things like that. And for that information, I will just put a few multiple choice questions on the final to cover that information. Fair enough, everyone? So does anyone have any questions? Yes.
So the 50 points of the pop genetics, that's just going to be in the final?
I'm thinking I'll just make the final out of 250 and not do a separate exam on population genetics. Okay? All right. Okay, so today what we're talking about is eukaryotic gene control. How do we control transcription of eukaryotic genes? And remember I told you they can be controlled at many levels, and we started off with regulation of the chromatin structure itself. And then we went on to, and I talked about the difference between cis-acting and trans-acting elements, and where we left off, the different kinds of RNA. And where we left off, I was talking about basal transcription factors. Now, remember with your basal transcription factors, if this is a gene, that's a piece of DNA, I'm sorry, and this is the gene of interest, what's this area here upstream of the gene? That would be what's always upstream of the coding sequence, let's put it that way, the promoter. And within the promoter, what's that special recognition site, the most important one? What's in the promoter? Who's talking to me? Very good, Carly, the Tatabox. And so remember with your basal transcription factors, the first one, and the one that's really, really, really important, is the Tatabox binding protein. It will recognize and bind the Tatabox, and then what happens to the DNA at that spot? It what? It bends the DNA at that spot. Okay, it seems to be that this has to happen first. So once we have the Tatabox binding protein on the DNA here, that's the Tatabox binding protein, then there are other transcription factors. They're called what? What are the other basal transcription factors called? Tatabox binding protein associated factors. That is correct. And how do we abbreviate that? TAFs. So Tatabox binding protein associated factors. So they're called TAFs, and they'll come, and of course they will bring with them RNA polymerase and align it up so it can transcribe the gene. Now, what you must understand is when you have the basal transcription factors loaded onto the promoter, this allows basal transcription of the gene. What do I mean when I say basal transcription of a gene? Only a small amount, right? So obviously we need some help if we want to have a higher level of transcription. And so the enormous range of gene regulation is due to a variety of other transcription factors that bind the enhancer region. Now, what can you tell me about an enhancer region? What can you tell me, folks? It is very far away from the gene of interest. So if this is my gene of interest, your enhancer element might be way down here somewhere. So there's other transcription factors who bind the enhancer part, that bind the enhancer part. They can either enhance or repress transcription. Now, the first ones we're going to talk about is we're going to talk about transcriptional activators. Transcriptional activators. So these will do what to transcription? Enhance or repress it? Enhance, because they're activators. That is correct. Now, what you must understand is this. Transcriptional activators have two binding domains. They have two binding domains. The first one is called a DNA binding domain. And that is the part that recognizes the enhancer region on the DNA. That is the part that recognizes the enhancer region on the DNA. So everyone understand, it has two distinctly separate domains. One is called a DNA binding domain, and that's what recognizes the enhancer region on the DNA. And then secondly, Stefan, does it matter if your screen is like, is it still recording what I'm saying? That's the only important thing, okay? The second domain is a transcription activator domain. And this is going to interact with the basal transcription factors. activator proteins have two very distinct domains. One of the domains is a DNA binding domain. That's going to recognize what, folks? The enhancer region on the DNA. And the other is a transcription activator domain that's going to recognize what? The basal transcription factors. It's all going to be clear in a minute, but we have to talk about all the parts first before we put it together. Now, I want to talk first strictly about this DNA binding domain. The DNA binding domains of activator proteins, that DNA binding domain, the one that's going to recognize the enhancer region, have characteristic shapes known as motifs. And I think I introduced that word motif before to you, right? Now remember what DNA is like. It kind of looks like my arm here. And so for things to bind onto it, they typically have to have two prongs like that. So they bind like this onto the DNA. Everyone kind of like putting this watch on, where it sort of clamps over the DNA. So that's why they tend to have these characteristic shapes that are going to give them that clamp-like advantage to get them onto the DNA. Now, there's three in particular. The first motif, or the first characteristic shape, is called a zinc finger. I love the names they give for things. I love these names. Now here's what a zinc finger looks like. We have, and remember, these are proteins. These are my activator proteins. This is the DNA binding domain of my activator protein. So they're made up of amino acids, right? Everyone? So we're going to have a zinc in the middle, like that. Kind of like this. And then here's what happens. So these circles just represent random amino acids. And that is exactly what a zinc finger looks like. So there are two cysteine amino acids, two histidine amino acids, and in the center, they're connected to a zinc ion. And zinc ions are Zn++ like that. Because of the polarity and other properties of the histidines and cysteines and their placement, what happens is this forces a loop. This forces a loop. And we call it an alpha helix, that particular loop. So that forces an alpha helix. It's all to do with the properties of those amino acids. Remember when I gave you your codon sheet, the initial one, it had them color coded for different properties, different polarities, different acidities, and that kind of thing. Having to do with those properties is what forces this alpha helical loop. Okay? Now, as you can see, sort of turn it this way, it could easily clamp onto the piece of DNA. Do you understand, everyone? All right, the next two are a little bit easier. The second type of motif is called a helix turn helix.
And this one has an alpha helix. It turns on an alpha helix, like that. Alpha helix, turn alpha helix.
Sounds like a dance step.
It does. And the third one is called a helix loop helix. Does everyone have these times and dates written down here? Everyone got these written down? Everyone? Okay. Helix, loop, helix. And again, we have our helix like this, and then it loops like this, and then you have a helix. All right? So, what I want you to understand is the DNA binding domain of the original activator protein. This domain right here typically will have one of these three motifs. So it doesn't look like this, it looks like one of those three, okay? And because we have the double pieces coming down, that makes it easy for it to bind onto the DNA at the enhancer site. So is everyone good with that? Everyone good? So on your exam, you can expect a question about what types of motifs are prevalent on the DNA binding domain of activator proteins. And they would be your zinc fingers, your helix turn helix, and your helix loop helix. Everyone good with that? And know how to write them out. All right, I have to erase this so I have room. But now, let's look at the transcription activator domain. This transcription activator domain, this is the part of that protein that's going to interact with the basal transcription factors. And most commonly for these domains, they don't have a characteristic motif like the DNA binding domain does, because they're not interacting with DNA, they're interacting with other proteins, the basal transcription factors. But what you do see on the transcription activator domain, that they are extremely rich in the amino acids asparagine or proline. Asparagine or proline. And these amino acids are acidic and that is what gives them their a tighter binding with the particular amino acids of the basal transcription factors. So just to sum this all up, this is an activator protein that is going to enhance transcription of a eukaryotic gene. It has two domains, a DNA binding domain that binds to that enhancer region way far down from the chromosome, way far down on the chromosome, and then a transcription activator domain that's going to interact with the basal transcription factors that sit on the promoter of the gene. Now, who's sneezing? Is it my friend? Oh, okay. There's no chalk in here. Where is that girl that sneezes all the time? There she is. You have glasses on today. Do you always wear glasses?
I don't like wearing them, so I usually don't.
I know, but you're so cute in glasses. It makes you look very intelligent. All right. Now, everyone, don't get confused with this next thing. In addition to this domain and this domain, there's a third domain. But the third domain is not on this activator protein. It is between this activator protein and a second activator protein. So, it's, okay, so let me finish and then you'll understand. When we do molecular analysis on activator proteins, it becomes very apparent that they work in pairs. So there's not just one activator protein, but there's two coming together and working as a pair. And so the third domain is not on one of the individual activator proteins, but rather it is right here between the two. Does that make sense? Yeah. Okay. Because activator proteins work in pairs. And this particular domain is called the dimerization domain. Can anyone think of why it's called that?
They make dimers.
Because they make a dimer, right? Two proteins together make a dimer. This dimerization domain is also characterized by a certain motif. This dimerization domain is also characterized by a certain motif. And you're going to love the name of this one. You're going to love this, guys. You're going to love it. It is called a leucine zipper. It is called a leucine zipper. So it appears that the way the proteins fold, they generate an alpha helix with these leucines sticking out like this from one protein, and an alpha helix on the other with leucines sticking out like that, and the leucines interdigitate with each other, like a zipper would when you zip it up. Does that make sense, everyone? Does that make sense? Got it? So it's just the way the proteins fold. They have this leucine sticking out like this on one, leucine sticking out like this on the other, and then they come together and interdigitate like that. And that's kind of like what happens on a zipper when you zip it up. And so we call it a leucine zipper. So what are the three domains we're worried about with activator proteins? Domain number one, up here is the DNA binding domain that recognizes and binds to what? The what? Enhancer region on the DNA. The second domain is the transcription activator domain that recognizes and binds to what? Basal transcription factors, or interacts with them. And the third domain is this new one here, and what's it called? Dimerization domain. Now, what are the three characteristic shapes of the DNA binding domain?
Finger,
Zinc finger,
helix, turn helix,
helix, turn helix,
helix, loop
helix, loop helix.
I thought that the enhancer was kind of far away from.
It is, and that's why this probably isn't making sense yet, but it's going to in about 1 second. Okay? And what is special about the transcription activator domain? What do we see in it? Asparagine and proline rich in those amino acids. And then what is special about the dimerization domain? The leucine zipper that holds those two activator proteins together.
and they function as a single unit. All right. Are we ready? Okay. Suppose this is my original chromosome. And suppose my gene of interest is right here, and here's my data box. So this is going to be looped up, right? Suppose my enhancer region is way down here. What is the only way I can bring that enhancer region within close proximity to that gene? Bending the DNA, exactly. And that's exactly what happens. So we're going to have DNA bending proteins that are going to bend the DNA like this around. So now there is my enhancer region. There is my enhancer region. So here are my bending proteins in here, and they will bend it around. Now, what protein is bound right here? Everybody, what protein is bound right there that caused the bend in the DNA? What? This is the promoter of my gene. This protein bound and it induced a bend in the DNA. What protein is that? Catabox binding protein. You are correct. Don't go away from that page because I'm going to ask you another question. What are these other proteins here associated with that? Those are your TAPs. That is correct. Now what we have is, if I was to draw something like this, what can you tell me that is?
Zinc.
It could be a zinc finger or a helix, turn helix, right? That's the DNA binding domain of your activator protein. Everyone with me on that? And how many activator proteins typically work together? Two. Okay. So this is the DNA binding domain of one of them. And this is the DNA binding domain of the other one. Everyone with me? And what am I drawing here? The leucine zipper, which is which domain? Dimerization domain. And I have to add a few more taps because I didn't make this long enough. But do you understand then? This becomes the transcription activator domain that associates with those basal transcription factors. Does everyone understand? Does everyone understand? Got it? Got it, got it? Sweetheart? You got it?
Yeah.
You're just looking it over. Make sure you get a good picture of this drawing, okay? Are you down on this? Okay. How about my ladies in the back row and gentlemen? Good? Everyone good with that? Okay. So, this is how eukaryotes activate or enhance transcription of their genes. Transcription is enhanced by these activator proteins. Yes, Joe?
So, in folding there, I mean, so the whole DNA is not being, there's a lot of folding going on there. So what, it just seems like, you know, for it all being connected and being tightly, if it's tightly bound,
It does seem like a lot to go along with that. But it doesn't.
Why'd you just put article polymerase up there?
I don't know what the answer to that is, but I just know that when it's in this position, RNA polymerase is constantly being re-recruited to this promoter, and you're just transcribing this gene like crazy. Stefan?
Does it kind of like make a wall that just like lets it come in?
Yep. It
So
does.
that's pretty much the signal. Like when that forms,
Mhm.
they know to keep
That
going
is
to
correct.
that spot.
And what it also does is it holds these. Remember, I told you that everything in the real world is always in motion, right? So these basal transcription factors are hopping on and off, on and off, on and off, and that's why you get very low transcription of them normally. But what does this do? This holds them on there and doesn't let them keep hopping off as frequently. Do you understand? And so that enhances transcription. Now, repression. It's just as important that we're able to repress transcription as that we're able to activate it. Now, I'm going to tell you a little secret. In prokaryotes, it's basically all about, is there a repressor sitting on that gene, or is it pulled off the gene, right? So repressors are really important. In eukaryotes, repressors are also really important. Now, a repressor is a protein that is designed to repress transcription of the gene, stop or slow down transcription of the gene, right? But in eukaryotes, here's the difference. Very rarely, once in a blue moon, which is 2 full moons in a month, just so you know what it is, will a repressor actually physically sit on the tata box and block the basal transcription factors. It can do that, but that's not the normal way that repressors work in eukaryotes. So instead of binding the DNA and physically blocking transcription, repressors tend to do something called quenching. They tend to operate more through quenching. Now, listen to me carefully. I don't mean for you to misunderstand this. Repressors can go and bind this tata box and block the tata box binding protein. And they can go and bind that enhancer region and block the DNA binding domain of the activator protein. But that's not their norm. Normally what they do is they do quenching. So quenching involves the repressor binding to the activator proteins. So here are my activator proteins, right? I'm going to try and draw them the same kind of way. And then we have these motifs up here, kind of like this. And then we have, and then this part. So what domain is this up here of my activator proteins?
Zinc finger.
Well, yeah, but what that's the DNA binding domain. What domain is this here? Dimerization domain. And what domain is down here? Transcription activator domain. So what quenching is when the repressor binds onto either the DNA binding domain, and that will prevent it from binding the enhancer region. Do you understand? Yeah, so the repressor is a different protein totally. And it will come in here, and it'll sit like this. And if it's sitting there, can this any longer go and bind the DNA? So they repress by blocking the activity of the activator protein. Does that make sense? Got that? Yeah. Little sweetie? Give me those genes next week. I'll put red patch on that.
Old Navy.
Old Navy?
They came with
I'm sure they did, but I can patch it for you.
Do we need to know, like, the purpose of blocking the DNA binding domain?
Well, yes, because that's how they block transcription. You understand that activator protein there, if it's going to enhance transcription, has to bind onto the enhancer region, right? And if it's blocked, can it any longer bind onto that enhancer region? No, and so instead of the repressor going and directly binding the DNA of the enhancer region, they typically will come and bind onto the activator protein and prevent it from binding the DNA. Does that make sense, everyone? Got it, everyone? Okay. Where's the other place they could bind? Where's the other place they could bind? And block transcription. What?
at the bottom.
At the bottom, exactly, the transcription activator domain. So they can bind the transcription activator domain just as easily. And if they bind this here, then it prevents them, they won't fit in there. They can't bind to the basal transcription factors. And that's called quenching. Now, typically, they don't bind in the dimerization domain. Can anyone take a guess why?
This is such a, it's such a tight fit as it is already. It's kind of hard for them to access
Well,
it.
but as the proteins are folding and leucine, they're coming together, one could sit in there, right? And prevent them from infiltrating. But we feel the reason it's kind of not as good to bind here is because guess what? It has to bind to one or the other. You still have two functional domains. So while it might not enhance transcription as much as normal, it would still be able to bind there and enhance transcription to a certain degree. So it's much more effective to block it completely. Okay, does everyone understand? Joe, yes. Well, I just said that at the beginning, right? I said repressors can sit directly on the enhancer or directly on the Tata box, but that's not as typical in eukaryotes. They're more likely to do this quenching instead. But that doesn't mean they never do that, okay? All right. Now, the dimers, these two activator proteins, the dimers, sometimes the two activator proteins are identical. So two identical proteins come together and form the leucine zipper. In that case, they're called homodimers. They're called homodimers. And the best characterized homodimer is one that is called June. And again, I didn't give them their names. Probably the guy who discovered its child's name was June or something.
That's
Okay?
Korean.
It's Korean? Jun.
Sounds like it.
Yes. All right. What do you think they're called if the two proteins making up the dimer are not identical? Heterodimers. That is correct. And this is going to throw a monkey wrench in your thought because these are called June phos. And I don't think phos is a Korean, is an Asian name, is it? June phos. That's the most common one. Now, take home question for exam number 3. I expect this to be handed in on the day of the regular exam, the day after Thanksgiving. And you're going to read about a different regulatory system, not a June or June phos. You're going to read about a different one that is called Max. Max. That's your heterodimer. And it's very interesting. I expect it to be typewritten. Stefan? Okay, I just wanted to make sure you weren't sleeping. I expect you to have some pictures. You'll need pictures to explain to me what's going on with the MICMAC regulatory system. This will be worth 20 points for your third exam. So the exam itself will be worth 130 points. Or no, let's make it 25. That's easier. Then the exam is 125 points. Okay? So I think that you've got lots of time to work on this, right? So today is Tuesday. Today's Thursday. You have about 10 days to work on it, including 2 weekends. Yes.
So what exactly do you want?
I want you to research and write in the paper about it. What is MICMAX? And how does it regulate gene expression? Right? How does it regulate gene expression? So does anyone have any questions about this part? Anyone have any questions?
You said write a paper on that.
MICMAX, yes.
How long do you want it?
Oh, 20 pages.
Oh,
You know what?
But just put relevant
Take it
I
information first.
refuse to set a length for any kind of assignment. Because if I tell you I want it 5 pages long, you'll write a good first 2 pages and everything else is just repeating what you've already written. And then it's boring for me to read. So, not boring, I don't mean it that way, but it's so repetitive. So, make it as long as it needs to be to explain to me what the MICMAC regulatory system is.
Do we need a reference page?
You do not. You can go look it up anywhere. Nope, in your textbook, should have some stuff on it. You can go online, I'm sure you can Google MICMAX, but don't take it from, what's it called? Wikipedia.
As far as that
Or
drawing
you
goes,
can
is
yourself
the,
too.
is there a name for that whole structure once everything's attached together?
No, these are your basal transcription
Yeah.
factors. These are your activator proteins. And the basal transcription factors are the ones that bring RNA polymerase in.
Yeah,
So
there's
they-
no name for that
There's
whole
no
thing
name
put
for the
together.
whole thing. That's just the activator complex, I guess. You could say. The whole thing. Stefan?
No, I was just.
Oh, you're just waving at me. I love it when you wave at me from the back of the room.
She did earlier. So
All
that's
right.
where RNA polymerases get a bind to it and for transcription. That's her.
Any questions? Okay. Now, hot off the press comes the next topic.
Ooh.
We've had control, right, with the histones. We've had control with transcription activation or repression. Now our pieces of mRNA are made. And what we're finding now, and this is recent. I graduated with my PhD in 2002. And they knew nothing about this at that time. So that's how recent this information is, right? There appear to be these little pieces of RNA, single-stranded pieces of RNA that float around the cell, but they don't encode for a protein. So for that reason, we call them non-coding RNAs or ncRNAs. They start out as a relatively large piece of RNA called the precursor, and then they are cut, and so they end up being around 22 base pairs long, which is very small, right? So these do not encode for proteins. They're single-stranded pieces of RNA in their mature form, that are about 22 nucleotides long. And so base pairs is totally wrong there. Someone should have caught that. They don't base pair with anything. They're single stranded. 22 nucleotides long. Is there music? That's so cool. We get serenaded now. In our
Oh, it's better. It's better than the construction.
It's-
Yeah.
yeah. Abraham, that came in just on cue. Now, here's the deal. There's different kinds of non-coding mRNAs. Some of them are called microRNAs, miRNAs. Some of them are called small interfering RNAs. My favorite are called peewee RNAs. Basically, the difference is in the precursor molecule that begins the whole thing. So micros come from one type of precursor molecule and the peewees come from a different. But here's how they work in general non-coding RNAs. They form a complex with a protein. So there's my little non-coding piece of RNA that forms a complex with a protein. And then they travel around the cell looking for mRNAs. Looking for mRNAs. How did you do that? You did it. Oh, God, it was straight to my hair. Oh, okay. I guess I'll never get it done. Now, these little protein ncRNA complexes travel around looking for mRNAs. And what does mRNA stand for? Messenger RNA. The messenger RNAs do, in fact, encode for proteins. Everyone with me? So these little ones don't, those ones do. And remember, it's all about controlling transcription, right? The mRNA is made already, and so if we don't want the protein from that, here's what happens. These little non-coding RNA protein complexes will come, and all they need is about 8 bases that they can recognize in succession and bind to, and they will bind right on to this piece of mRNA. So there's my little non-coding RNA.
That would be nucleotide.
What?
Nucleotide.
Oh yeah, they would be nucleotides. Whatever. No, I'm teasing you. So they have to have about 8 nucleotides that they can base pair with out of the 22 or so that they are long. So there would be on the mRNA, let's say there's an A, U, C, 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 and on this little piece of non-coding RNA, there's a U, A, G, 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 G. Then it can bind. Do you understand? Everyone. And then guess what? What has it done to that piece of RNA? What has it done to that piece of RNA? Blocked it. You can't transcribe it. And sometimes that's all it does. It sits on there and just blocks it. But other times it sits on there and actually then the protein will degrade that piece of RNA. So these non-coding RNAs are something that is being studied a lot right now.
Which portion gets degraded? mRNA or
The mRNA will be degraded. Yes. Yes.
You said MI is micro and SI is small. Interfering.
Interfering.
What's
Peewee.
peewee?
I don't know. That's the whole name of it. Peewee.
It's just
P-I-W-I.
it doesn't stand for anything.
After Pee Wee Herman.
Ah.
No, maybe. I don't know. I just made that up totally. But we think of peewee as being tiny, right? And so I'm sure that's where the name came from. I like to think it was from Pee Wee Herman, though. Just makes it more fun. All right, another way we can control transcription of specific genes is through something called alternative splicing. And this is really cool. Alternative splicing means the piece of mRNA is from a single gene is spliced differently. So you have one gene, you make two pieces of mRNA, and they're spliced differently. And so you generate two different but related proteins.
Can you repeat that, please?
Yes, so you have one gene, you make two pieces of mRNA from it, which should be identical, but they're spliced differently. So you remove different nucleotides from it. And then you end up with two different but related proteins. Now here's a really cool example. Follow this through because you might see this again. In Drosophila, females during embryonic development, they produce a protein called SX1. So this is females during embryonic development. They produce this protein. It is essential for female development. Male embryos do not produce this protein. And so how does it work? Well, here's the gene for SX, and upstream of it is not one but two promoters. And they're labeled PE for promoter early and PL for promoter late. Now, during early embryonic development, the female flies, female embryos, make transcription factors that recognize the early promoter. And will allow them to transcribe this gene. Okay, so during early embryonic development, females make transcription factors that recognize just this early promoter. Males do not make them. Later on in embryonic development, these transcription factors for this early promoter disappear. So we can no longer transcribe from that promoter. So we have to transcribe from the late promoter. And females continue to need this protein. So we must continue to transcribe from that gene. Now here's the clinch. Once we start using the late promoter, we find it is activated in both females and males. We find it is activated in both females and males. But we can't let the males make that protein, or they'll turn into females. Well, that wouldn't be good. But so as that you're not confused. Females, early in embryonic development, make transcription factors that transcribe off this early promoter and allow them to make this protein. Males do not make transcription factors that recognize this. So early embryonic development, males are fine. They don't have to worry about it. But later in embryonic development, the transcription factors for this promoter are turned off. They're no longer made. So now they're forced to transcribe from this late promoter. And males have that late promoter and make the transcription factors that recognize it. But the problem is, we absolutely need to continue to make this protein for females, and we absolutely cannot make it for males. Okay? Everyone got me? All right, so what happens? Well, here's what happens. Once the mRNA transcript is made, it is spliced differently. And when we splice the males, we leave in one exon that we take out when we splice it for females. So when we splice it for a male, we leave in this particular exon. So in the females, this exon gets removed. In the males, this exon stays in. So it becomes, no, wait a minute. Exons stay in, yes, that's right. So the exon stays in. So what I'm trying to tell you is, in females then, what is this? It's no longer an exon, it is an intron, and it's spliced out. And guess what that exon encodes for? This is my mRNA, so I made my mRNA in both the male and the female, and they're identical, but the only difference is that in females, this little section is spliced out as if it's an intron, whereas in males, males, this little section is kept in. Okay, becomes an exon. So in females, it actually becomes an intron. In males, it's an exon. Now remember, let's put this all together. Females want the protein made. Males do not want this protein made. We make this protein in males and they're gone. Okay? So what do you suppose that exon encodes for?
Stop.
A stop codon. Exactly. And so now the ribosome sits on the mRNA, transcribes along, and very soon runs into this stop codon here. You understand, everyone? And falls off and doesn't make a functional protein from it. But the stop codon is cut out in the females. Does everyone understand? And that seems to be in eukaryotes, particularly humans. When we first did the human genome, we expected there to be a lot more genes than we really have, a lot more genes. And what we now know is that at least 75% of our genes have alternative splicing. So we're able to make more than one different protein from each of our genes. And we think that's why we can have so many proteins and not as many genes as we originally thought. Okay? So alternative splicing is going to be really important. Finally, we can control transcription by controlling how stable the mRNAs are. Now remember, when an mRNA is made, we put on a cap up here, right? And what do we put on the other end? Poly A tail. Poly A tails and the cap prevent the mRNA from being degraded. As soon as that mRNA is made, there are proteins that are going to start degrading it. The cap prevents it from that end, and they'll start eating away at the poly A tail from this end. So do you understand, the longer the tail, the longer it takes to degrade that piece of mRNA. And poly A tails literally range from 20 A's to over 200 A's long. And that's another big way we control transcription, by controlling how long that mRNA remains in the cell. One other way is called mRNA editing. We don't know how this works, but it appears that you make an original piece of mRNA, and then somehow specific codons get changed to different codons. We seriously don't know how it's done. We don't know what is doing it. So I like to mention this because this could be your future PhD work. Any of you in here could work on mRNA editing in your PhD. So now, what we have is transcriptions complete. Those are all the ways we can control it at the transcription level. The final level of control of whether you make a protein or not is protein modifications after they're actually made. So just because I make a protein doesn't mean that protein is going to be functional. Do you all understand? Just because we make one, it doesn't mean it's going to be functional. And it's probably a good thing we switch the exam because I don't have time to talk about protein modifications, but it's very sweet, short, and simple, so there won't be hardly anything left for this third exam. And as soon as we talk about protein modifications, then that will be it for this next exam. All right, everyone, good job. And I will get in touch with distance learning and make sure they check in on
Hey.
Yes.
On your mRNA editing, it appears you make a piece of mRNA